Hello, Tam here. This is Lesson 1, Sexual Reproduction in Plants. So learning objectives for today are to draw and describe the basic structure of a flower, describe the meaning of pollination, and then we'll look at the different types, so wind versus insects and self versus cross. And then also to describe the structure and development of pollen grains and ovules, outlining the role of mitosis and meiosis in each. So these are the spec points that we're looking at today. You might just want to pause the video and look at those in more detail. Okay, so please pause the video and complete activity 1. And just as a little thing, you might remember this very gender stereotype saying, so men stay men and have all the anthers, women have style and go to the toilet in carpels. So that actually helps us identify the male and female parts of a flower. So here is a cross section of a flower and just a few things to point out. So the first thing is obviously all flowers have petals, and in this insect pollinated flower you can see the petals are actually coloured, and that's to attract the insects, but all of the petals together are called the corolla. We've got the male parts of this flower. So remember, the flower is the reproductive organ of a plant, and so the male part of this is the stamen. And that's made up of a filament and an anther. And the anther is where the pollen is made. And then we've got the female part called the carpel. And there's a few bits here. So down here we've got the ovary. And later on we'll look at how this develops into fruit and seeds. Within the ovary we've got the ovules that will actually turn into the seeds. We've got the stigma at the top here, and the stigma is where the pollen will land, depending on if it's cross or self-pollination. We'll look at where the other pollen comes from. And then we've got the style, which is involved in double fertilization. We'll come back to that later. And that basically has the stigma attached on top. A couple of other bits actually to mention. So before the flower has bloomed, it will be in a little bud, and it will be encased in the calyx, and the calyx is made up of these sepal, and that basically protects the flower before it's bloomed. Okay, so please pause the video and complete activity 2. And just to review this then, so pollination is basically the transfer of pollen from the anther of a flower to the stigma of a flower. Which flower is insect and which is wind pollinated? So we've got a couple of pictures here, and you might identify straight away that this is the insect pollinated flower. So I'll just put a little bee on here. Okay. And this here is the wind pollinated flower. So a couple of things that you will notice. So the first one is the colour of the petals. So here you can see in the insect pollinated flower we've got beautiful colours, or we find beautiful colours, and that's to attract insects to the flower, whereas in the wind pollinated flower you can see that the petals are actually green or sometimes absent. The anthers and stigma, so within a wind pollinated flower you can see that the anthers and also the stigma, which are up here, hang kind of outside of the flower, and that's because obviously the anthers is where you have pollen, so hopefully the wind will pick up the pollen. It's more likely to if the anthers hanging outside of the flower, and then hopefully the wind will blow pollen into to land on the stigma, and again if that's hanging outside of the flower then it's more likely to land on it. Another thing is the scentedness and nectar. So there's no scent and no nectar in a wind pollinated flower, whereas in an insect pollinated flower, again to attract the insects, there is a smell and also the sugary nectar available. Larger versus smaller anthers. So again, you can't really see because these aren't sort of size comparable, but the anthers in wind pollinated flowers are much bigger compared to those of insect pollinated flowers. You also have larger quantities of smooth pollen in wind pollinated flowers, and so again that's because if you've got more pollen it's more likely to land on the stigma of another flower of the same species, whereas with insect pollinated flowers you don't need as much, and also the pollen is slightly different in how it's structured. So in insect pollinated flowers you'll find that the pollen is sort of stickier. It's got these little spikes on. You can see that up there, and that's to stick onto the insect. Whereas in wind pollinated flowers, you have smoother pollen, sometimes dappled actually, kind of like a golf ball, so that it will travel further in the wind. And then the final one is again looking at the shape of the stigma. So you can't really see it here, but sometimes, well, you can on the wind pollinated one. You can see we've got these feathery structures. Again, it increases surface area, so you get more pollen landing on them. Whereas in the smooth stigmas on the insect pollinated flower, again, you don't need a larger surface area because the insect's going to hopefully deposit pollen as it travels down to collect the nectar. So self and cross-pollination, you need to be able to define both of these. So self-pollination is where you get the transfer of pollen from an anther of a flower to the stigma of a flower on the same plant. And that's a really important thing to say, especially in exam questions. Cross-pollination, so the transfer of pollen from the anther of a flower to the stigma of a flower on a different plant. Okay, so again, just be really careful with those final bits when you're in the exam. An advantage and disadvantage to self-pollination. So an advantage here is that if the plant has a successful genome for its environment, that will be carried through to the next generation. But the problem is that if the environment changes, then the offspring will have the same genome available. So there's not going to be much genetic variation, which means that if you have a change in the environment, that particular offspring might not be very well suited to that environment. So the only things that are going to generate genetic variation are going to be things like crossing over, mutations, or independent assortment. Another thing is basically inbreeding. So it's reproducing with itself. So you're more likely to have two harmful or recessive alleles combining, producing a non-beneficial kind of phenotype in that offspring. Suggest how dichogamy and the structure of the flower up here may help reduce self-pollination. So a couple of things to mention. Dichogamy is basically where you have ripening of the stamen and pistils of the flower at different times. So basically the male and female parts mature at different points in time. So what that means is that the pollen, for example, couldn't self-fertilize the female gametes because the female part is not yet ready, maybe for reproduction, or vice versa. Protandry, so this, if you break that word down, actually pro means sort of first, andry, talking about male. So the male reproductive organs mature before the female. And you have progyny, which is the reverse. And then if we look at this flower up here, so things that reduce the likeliness that there'll be self-pollination is that the anthers are actually below the stigma, so it's less likely that pollen's just going to fall onto the stigma. So we're going to talk through this process, and then I'm going to get you guys to do a few questions on things. So the first thing here is if we take a cross-section of the anther, so remember the anther is part of the stamen, so the male part, and we're going to take a cross-section through the anther. So we can see that here, and we can see actually if we were to look down on this part here, it would look like this image. So we'll hopefully at some point get to look at these under microscopes, look at anther microscope slides. And so we really are interested in these parts here, the pollen sacs, okay? And this is where pollen is made. And so the process of how a pollen grain is made and matures is this. You have a microsporocyte. So microspores and megaspores are kind of terms for the male and female reproductive gametes in plants. So micro means smaller. As you know, male gametes through sexual evolution are smaller than female. So sperm, for example, is much, much smaller than an egg. So here, the microsporocyte, microspore, small spore, so it's smaller than the female one, which is called a megaspore. We'll come back to that in a second. So the microsporocyte divides by meiosis, and it divides into 4 cells called a tetrad. And these 4 cells separate, and each one of these is going to become its own pollen grain. And if we were to take a cross-section through those and look at what's inside, it would look like this, okay? And so here we've got a few things just to notice. The first one is we've got a generative nucleus. Now the generative nucleus is a haploid nucleus, so just N, and we've also got a pollen tube nucleus, which is also haploid, so again just N. These nuclei are genetically identical because this process here, from meiosis to this point, so this bit here, we actually have mitosis taking place, and mitosis has produced two genetically identical nuclei, okay, and they're both haploid, and you can see that here. So a couple of other things to mention. We've also got these little pits. So these are little holes in the outside part of the pollen, and this allows the pollen tube to grow later, and we'll look at that when we look at double fertilization. We've also got two layers of the outside coat of the pollen grain. So we've got an exine, which is a thick outer wall, and we've also got an intine, a thin inner wall, and these layers help stop the pollen become dehydrated. So pollen might be traveling in the air for a long time until it lands on a stigma, or it might it might have landed somewhere and potentially get blown off again, but they might be hanging around for a long time before they fertilize another flower. So they need to have a water resistant layer to stop them from getting dehydrated. Okay, so let's look at how an ovule develops. So we're going to start off with a megaspore, and this megaspore, this cell, is going to divide by meiosis to produce 4 haploid cells, and 3 of those haploid cells are going to break down, and one of them is going to grow into a much larger cell with a much larger cytoplasm. And you'll notice that's quite similar actually in human reproduction as well. The female cell grows very, very large and has a very large cytoplasm compared to the male gamete, the sperm, which is much, much smaller. So what happens next is we have 3 cell divisions, and these cell divisions are mitosis. So that nucleus is going to divide once, twice, and a third time to produce 8 nuclei. Now 3 of those nuclei will separate into their own individual cells called antipodal cells, and they'll be at one end of that embryo sac, and 2 of them will develop into the synergids. 1 will develop into the oosphere, so this is the female gamete, and then we've got these 2 left over in the middle, and these 2 here are going to fuse together. And if you remember that all of the nuclei in here at the moment are haploid, but these 2 will fuse together and they will produce a diploid polar nucleus. And that is how the embryo sac develops. Please pause the video and complete activity 3. So here, a few things that we should find. The names of the 2 nuclei within a pollen grain. So remember that in a pollen grain you have the generative nucleus, so this is the male gamete that's going to be involved in double fertilization later. We also have the tube nucleus. We're going to come back to this in double fertilization, so this helps with the pollen tube growth down the style. Dehiscence, so this is basically where the anther ruptures and the pollen is released, and hopefully we'll get to look at again cross sections of anthers and we can identify the exact point where this happens. A diagram to show a pollen grain in detail, so again you should have something like this, so just check that yours matches, and then a diagram to show mature ovules, so again just check with this diagram, see that you've got all the labels. Please pause the video and complete activity 4. Finally, there's a little Quizlet for you to try just as a quick summary. And just a quick review of all of the learning objectives.